Okay, well, whilst we just give Caitlin a couple of extra minutes to finalize the setup of a talk, I just welcome everybody to the first of this year's Fungisphere talks. We've had a little bit of a break, but we're hoping to have talks at least every month, and it's great to see so many people here. We are recording the talks and they'll be available on the website later. So we actually have two talks this month, but the first of those is coming from our clinical and translational theme. And Sharon is hopefully going to be connected and will introduce our speaker. So thank you.
Okay. Thank you, Justin. Good afternoon, everyone. I know you can't see me, but I work at the ICPMR, and for those that work in this, illustrious institution, you know that nothing usually works here, and there's no visual card on my computer. So you have to make do with my voice. So I have the pleasure of introducing Dr. Caitlin, who is doing her PhD with in mycology at Westmead Hospital in conjunction with Vitaly Wieland and Sebastian Van Houw. And today she will be talking about lessons learned in Candida tropicalis genomics. Over to you, Caitlin.
Thank you very much for the invitation to present. I've been really excited to have this opportunity, which has been rare with COVID. I'm presenting some work which forms part of my PhD. Thank you immensely for your patience once again, and I'd like to start by recognising the traditional landowners wherever you are. I'm going to have to scroll because I'm working with a PDF here. I'd like to acknowledge especially Dr. Marie Gore, who you heard just now, who's put a lot of effort into this, principal co-author and investigator, as well as my other co-authors, and the in-kind support from CIDM, Pathology West, Australian Society for Antimicrobials, for the research grant, and the Royal College of Pathologists Australia similarly. So what I'm going to talk about is the importance of Candida tropicalis, azole resistance and global sequence types, antifungal resistance to fluconazole in Candida tropicalis, and an approach to genomic analysis in a diploid organism. So, Candida tropicalis and candidemia. What you can see there is an image that comes from an Australian candidemia study, the most recent one, the ACS, and it shows the proportion of isolates causing candidemia, and 4.4% of them are Candida tropicalis. The mortality over the two Australian candidemia studies that have been done, separated by about a decade, hasn't changed, so it's remained around 30%. And Candida tropicalis candidemia has a higher mortality of between 37 and 60%. And the prompt for this study was the finding that resistance had increased from less than 2% to around 17% in the most recent surveillance study. Candida tropicalis is transmissible with various clusters described. I've picked out two recent reports. The top article in Mycoses last year from Serbia and the UK described two AML patients with candidemia that were separated by 10 days, and the bottom one described 9 isolates from candidemic neurological patients in the same ward, linked to the same isolate from the hands of a social worker by MLST. And there are at least 10 published outbreaks of clusters of C. tropicalis going back to 1989 and the cluster of sternal wound infections. MLST is commonly used to assess relatedness in Candida tropicalis. It has excellent interlaboratory reproducibility, and its advantages include the fact that it's so simple. It's highly transferable because all you need to do is transfer 6 numbers, a number for each allele. It's simple because it's greatly simplified. So there's less resolution than if you use whole genome sequencing data. And because of this, it's likely to be less accurate. So an example is if you have, for example, 2 SNPs in one allele, that allele will be given one different number. And that will be counted very differently to if you have one SNP, but those in 2 different alleles, so same number of SNPs, but in two different alleles, we get two different numbers for the different alleles, and that makes a difference of whether it is classified in the same clonal complex. The additional complexity when using it on a diploid organism is the fact that you can get heterozygosity, and so if there is a heterozygous site, you can assign an ambiguity code, and that gets assigned a different allele again. This is the MLST scheme for Candida tropicalis that was established in 2005 by Ariana Cervanti and Frank Odds, who unfortunately passed away last year. It's now curated by Sujung Lo, and the 6 housekeeping genes included are listed. It's quite small, so data from 1,253 isolates is included compared to 4,464 for the nearest relative Candida albicans or 35,000 for Staph aureus. Diploid sequence type is a term in relation to MLST that we use for a diploid organism, and that's because there is that additional factor of an ambiguity code where there is a heterozygous site. And the majority of studies have suggested clonal clustering of fluconazole susceptibility with diploid sequence type. This is an example of a paper from Thailand where they found that 10 isolates from blood culture positive cases clustered in a clonal complex. And there's a more recent paper a few months ago from China that also demonstrated clustering of fluconazole-resistant isolates, and they made a special mention of clonal complex 2 with diploid sequence type 225 being highly associated. What are the mechanisms of azole resistance? This is a slide from a review last year, which I'll take you through. So azoles are fungostatic, and they work by targeting the lanosterol 14-alpha demethylase, which is coded by the ERG11 gene, which thereby inhibits ergosterol synthesis. So the DNA picture at the bottom is meant to represent mutations that can occur in ERG11 that decrease the affinity of the drug for the target molecule. And those mutations occur spontaneously. ERG11 expression is under the control of UPC2, which can upregulate its production. Now, there are 2 families of transporter pumps that can pump out the azole. So they are the ATP binding cassette, or ABC pumps. They are under the regulation of TAC1, and the major facilitator superfamily, MFS, which are under the regulation of MMR1. These can be upregulated and commonly are so under selection pressure. These changes are inducible in vitro. Now, the genetic mutations possible in a diploid yeast include amino acid substitutions, but also include these structural variations. So you can get aneuploidy. You can also get copy number variation. So that is where you get an increased copy number or a different copy number of a particular gene. And you can get loss of heterozygosity, where you can get either haploinsufficiency or haploproficiency. And I'm highlighting C and B and loss of heterozygosity because they have been associated with azole resistance. So our objectives were to place Australian isolates into the global context of available azole-susceptible and resistant isolates, with the hypothesis that Australian isolates will be found to be diverse and disparate, and there will be an association of MLST with azole resistance. Secondly, to identify mutations associated with antifungal resistance using whole genome sequencing, and with the hypothesis that isolates resistant to azoles will have mutations clustered in the ERG11 gene, and that novel associations may be uncovered. So we had 69 Australian bloodstream isolates that were included. There were 10 isolates contributed by a group in Singapore, and we also included the ACCC strain. We were able to download 4 sequences from the NCBI database from Canada. Susceptibility testing was done using sensitivity yeast 1, DNA extraction was done using the MasterPure yeast DNA kit. Genomics libraries constructed using the NextEra kit, and sequencing was performed on the Illumina NextSect platform with FastQ quality evaluation using FastQC. Mapped trim reads were mapped to the reference, the annotated reference, using BWA-MEM, and variants were predicted using VARSCAN and then annotating using SNP-F. The website that I showed was used to assign MLST, and the relationships between diploid sequence types was established using an eBurst algorithm and PhyloViz. Phylogenetic tree was constructed from consensus sequence generated using BCF tools. A curated list of resistance genes were generated from the literature, including a limited number from a database called MARDI, and variants that were discovered using VARSCAN were filtered for a curated gene set by excluding SNPs present in susceptible isolates. Copy number CNV changes were characterized using CNV NADA across the ERG11 gene, which is the one that's been associated with azole resistance, and regions of loss of heterozygosity in ERG11, TAC1 and MMR1 were investigated. So they're the regions that have been associated with azole resistance, ERG11 and TAC1 being right next to each other on chromosome 5. So we had 84 isolates with the breakdown as listed. Importantly, 17 fluconazole non-susceptible isolates and a large cohort, which is one of the advantages of the study, 67 fluconazole susceptible isolates. This is an eBurst that demonstrates, as suspected, the Australian isolates looked dispersed and disparate. And this is an eBurst that demonstrates fluconazole susceptibility. So in red, non-susceptible, green, susceptible, and in pale yellow, unknown. And that clonal complex 2 diploid sequence type 225 that I'd mentioned had entirely resistant isolates that included a Melbourne isolate as well as Singaporean isolates. This is a list of the 17 fluconazole non-susceptible isolates in order of fluconazole MIC. So across the top, you've got the drugs tested in susceptibility testing, and in the boxes are the MIC. So you can see the fluconazole MIC is in order from greater than 256 down to 4, and it's shaded according to the cutoffs in CLSI. So red being resistant, green being susceptible, light orange being sensitive dose dependent, and the darker orange being above the epidemiological cutoff value because there hasn't been a formal breakpoint assigned. And in the last column are the amino acid substitutions that were found in the ERG11 gene, were not present in susceptible isolates. And the thing that you'll notice, so in bold are the ones that were found in the homozygous state, and in not bold are those that were present in the heterozygous state. Where there were 2 homozygous amino acid substitutions, there was a higher MIC than where there were either 2 heterozygous mutations or a single homozygous mutation. So ERG11 mutations were present in the majority of the non-susceptible isolates. That left 6 isolates that didn't have an explanation, and this is a different representation of the same kind of diagram as a reminder that the other places we could look included some of the genes mentioned, the ABC transporters, the MFS transporters, and TAC1, MMR1, UPC2, which induce their expression, or UPC2 induces ERG11 expression, as well as NDT80 and STB5 as inducers of ABC expression. And finally, copy number variation and loss of heterozygosity as structural features. So how did we look at these genes? Well, this is a list of the genes that we looked at, and there's another slide where it has a description, so don't worry too much about the list. This Venn diagram is a representation of all the amino acid substitutions that were found. So there were 347, and then it was a matter of figuring out which ones were exclusive to the non-susceptible isolates. So in dark green are the mutations that were present in the homozygous form in fluconazole susceptible isolates. In light green, those that were present in the heterozygous form in fluconazole susceptible isolates. In dark red, those that were present in the homozygous form in fluconazole non-susceptible isolates. And in light red, the ones that were present in the heterozygous form in fluconazole non-susceptible isolates. And it's really the ones that are present in the red, and particularly in the dark red and not present in the green, that are of interest to us. At the bottom is a little bar graph, and the thing that your eye was probably drawn to is the high rate of background variation. So the majority of these changes were found in heterozygous form in fluconazole susceptible isolates, representing that background variation. So honing in on those changes that were present only in the red parts of the Venn diagram, in the resistant isolates only. This is a list of the genes, their description, the amino acid substitution, and the frequency of finding it in the homozygous form and in the heterozygous form. So you can see that the ERG11 mutations were, as I mentioned, the predominant. They are the ones highlighted are the ones that have been described previously in the literature, and they've been proven to cause azole resistance by taking these and transfecting them into a Saccharomyces cerevisiae and then demonstrating that they caused a substantial rise in the fluconazole MIC. Now, there are only 2 other genes that had homozygous mutations, and you can see that the frequency was 6%, which represents 1 isolate. They're in a CDR11 transporter and STB5, and then the remainder were found in the heterozygous form. So this is a maximum likelihood tree with a heat map of candidate amino acid substitutions, as well as the CNV and ERG11 and loss of heterozygosity. And don't worry too much, I'll zoom in on particular bits. But just to start with, to highlight this little group here, which I've already mentioned, with Singapore isolates and a Melbourne isolate. The red there represents the fact that they're fluconazole resistant, and the pink represents the homozygous mutations noted in the ERG11 gene. So, honing in, you can see here that the along the top are the gene and the amino acid substitutions, and then it's coloured in if it was present in that isolate. And you can see that the STB5 homozygous mutations were present in isolates that already had an explanation for their raised MIC, as were the heterozygous mutations in the TAC1 gene. So whether or not these were causative or additive is unclear, because they already had a good explanation. So then if we just focus in on the isolates that didn't have an explanation, which were those 6, and just list the amino acid substitutions that were present in them but not in susceptible isolates. In this isolate from Sydney, there was a heterozygous amino acid substitution found in TPO3, which is a transporter pump. Now, that may or may not be instrumental. I suppose we won't know until it's tested, but it possibly isn't, because you would have to have a mutation that causes the pump to work more efficiently at pumping out azoles, and also be expressed even though it's only present in the heterozygous form. This Canadian isolate that had been downloaded had a CNV in ERG11, equivalent to a copy number of about 6, and that had been previously reported as not present in a paired isolate that was susceptible and isolated prior to that time point in the same patient. So that was thought to be the mechanism for that MIC of 32. And then the remaining isolates, we've got a loss of heterozygosity in MMR1, and amino acids, heterozygous amino acid substitution in an transporter pump, other transporter pumps, or heterozygous amino acid substitutions, and there is that homozygous amino acid substitution in a transporter pump. Now, the other thing to note here is that these ones that were otherwise unexplained tended to have a lower MIC, and 4 is actually susceptible dose dependent. So although this is reproducible, it's within the margin of error of the MIC being 1 to 2 dilutions, that it could actually have been phenotypically susceptible. So what about this CNV in ERG11 and the loss of heterozygosity in ERG11/TAC1 or MMR1? That has been associated with azole resistance in the literature, as I mentioned, but here we have a large susceptible cohort. So just on this bit that's zoomed in, you can see that already that's at the end, you can see ERG11 in CNV shaded in a lighter blue for copy number closer to the diploid state, and in darker blue, higher copy number. And then in the last 2 columns, shaded if there was loss of heterozygosity present in the MMR1, and then finally in the TAC1 and ERG11 genes. You can see that those are present in susceptible isolates as well as in resistant isolates, and there's a table there just to demonstrate that point, that it was not strongly associated with fluconazole non-susceptibility, and indeed this would suggest that it's not sufficient in and of itself to cause azole resistance. And I suppose that's probably because gene dosage is not the whole story. So just because you have an increased copy number doesn't mean that it will be transcribed, that there will be RNA expression that then leads on to a phenotype. This is from a paper looking at Candida albicans, and this patient 43 in green demonstrates that it had a trisomic chromosome 5, so 3 copies of that chromosome 5, an area of loss of heterozygosity there, and yet that isolate had an fluconazole MIC of 0.75. And it has been reported that RNA expression levels don't always correlate with gene copy number. There's phenomenon of dosage compensation that can occur. So back to these. Oh, the other thing I wanted to mention here actually is that with these structural variations, they can be induced in vitro when you passage the isolate in the presence of fluconazole, and they probably have a cost of fitness to the organism, so they can be lost in the absence of that fluconazole pressure, and they're not stable features. A lot of studies. So moving then to ERG11 mutations, and what you can see here is that the ERG11 mutations were present in independent phylogenetic branches and are present across this cluster. And ERG11 mutations, I mentioned, occur spontaneously, and it's rarely possible to induce these in vitro. They probably occur on a bigger scale, and then those organisms are selected for. And so that brings the question of where do these organisms come from? And it's been a question of study for this group in Taiwan, and they found that clonal complex 2, diploid sequence up 225 on Candida tropicalis that was resistant to azoles on fruits that were found from the supermarket, and also in human isolates and soil isolates, suggesting that potentially it's agricultural azole use that is providing the pressure. And so that leads into this kind of One Health idea that, azole use in humans is really important for prophylaxis and treatment, and it's being impacted possibly by agricultural azole fungicides as drivers of antifungal resistance in pathogens of humans, and particularly in Southeast Asia. So the question arises whether that Melbourne isolate, maybe it was related to travel, but certainly antifungal resistance, the rise of antifungal resistance may well be related to global travel. Maybe that will be less of an issue with the recent year that we've had in COVID. So we're ready to use this approach to look more broadly. Well, in terms of implementation and translation of whole genome sequencing, there are challenges. It's probably not ready for prime time. It's very expensive and time-consuming. There are some gaps in mycology and genomics that have, I suppose, been addressed in a bigger way in bacteriology and virology. So the antifungal resistance databases that exist are quite limited, and there's a limited availability of benchmarking data, as you can see from the number of isolates we were able to download. And maybe in the future, phylogenetic analysis will supersede MLST because of its greater resolution and greater flexibility, and RNA sequencing would allow greater understanding of gene translation. And this is a slide to illustrate some of the challenges presented by doing genomics in a diploid yeast. So a comparison of Candida tropicalis and E. coli. Candida tropicalis is diploid and has 6 chromosomes, whereas E. coli has a single chromosome. Candida tropicalis is large, 14.5 megabytes compared to E. coli, which is a lot smaller, and that translates into needing a lot of space for data storage, which can in itself be expensive and means that it's not easy to transfer this data because of upload speeds and internet. Today is an example of the importance of upload speeds and internet. It has predominantly clonal reproduction but can do odd things, and the reference genome is annotated to supercontigs, whereas for E. coli you've got established drug resistance databases and online pipelines. So I suppose with whole genome sequencing, there are vast amounts of data. It can be complicated fishing out the relevant information. Just because there's a lot of data doesn't mean there are a lot of answers. So in summary, Candida tropicalis carries a high burden of disease. MLST analysis demonstrated diverse Candida tropicalis population with clonal clustering of azole-resistant isolates. Candida tropicalis resistance to azoles is increasing and is often mediated by SNPs in the ERG11 gene. Genomics in eukaryotes is complicated by multiple copies of each chromosome and by plasticity of the organism, and antifungal resistance in Candida tropicalis may be yet another illustration of the impact of global travel. Thank you very much to my co-authors and to my audience again for persisting. And I'd like to invite questions.
Thank you very much, Caitlin. That's an excellent talk of everything. Maybe if you'd stop sharing your screen, then we can get you back in the picture for answering questions. Okay, fantastic. Just change this view. Okay, so do we have anyone with any questions for Caitlin? Who would like to go first? You can feel free to raise your hand or just unmute yourself and talk.
I'll start with a question. Thanks, Caitlin. It's Julie Georgievic from SIDM at Westmead. Thanks for the talk. Look, I might have missed it at the beginning, but the isolates that you were looking at, were they a combination of environmental isolates as well as clinical isolates? Because I guess I'm just wondering about how much inherent azole resistance there is versus resistance that's developed as a consequence of treating the patients.
Thanks for the question, Julie, and that raises a point that I wanted to raise. So they're all blood culture isolates. They're all from patients who had candidemia. The Australian isolates are basically all the ones that we could include and had access to. So they represent the largely the first isolate in a candidemic patient without necessarily selecting for azole resistance. The Singaporean isolates, by contrast, were mostly azole-resistant isolates, so 7 of the 10 were azole-resistant. And there could certainly have been sampling bias in those Singaporean isolates. We don't have any details about the clinical context and whether they were linked epidemiologically, for example.
Okay, thanks.
Do we have any?
We can't hear you, Justin.
I know, sorry, I just noticed the mute. Does anybody have any further questions for Caitlin?
Caitlin, it's Wieland.
Hi, Wieland.
Very nice talk. Did you, when I listened to the last answer, so do you plan to look at some of the do you have some serial isolates of patients which are treated, and you see if there is anything coming up to develop resistance after treatment? Just to follow up on Julie's question also a little bit. So do you plan, you mentioned that the Australian isolate was the first isolate from a patient. So are there isolates which have been obtained in a row afterward when the treatment started or during treatment, or do you plan to follow this up in this direction?
Mm-hmm. Thanks for the question. The short answer is no, we don't have those isolates.
Okay.
Yeah. So they're single isolates.
Mm-hmm.
And in terms of whether they're likely to have been pre-treated, we don't have data on fluconazole prophylaxis or prior treatment data. That would be really useful, but it's entirely a laboratory study.
Mm-hmm.
The Canadian isolate would be the exception. So there were 2 isolates that we downloaded, or 2 sequence that we downloaded sequence data for 2 isolates from a Canadian group where they'd had a patient that had a susceptible Candida tropicalis peritoneal isolate and then had several months of therapy, and then had a resistant Candida tropicalis isolate from the same site. So the MIC had gone from 2 to 32, and that was the one that the only difference they could find that might explain it was that CNV in the ERG11 gene.
Mm-hmm.
So that supports the idea that under selection pressure, an isolate can have these structural changes that will help it adapt.
Mm-hmm.
But the bulk of the data would suggest that happens under fluconazole selection pressure, but it's not stable. So it's not the kind of thing that is, I would think, that I would think would be as transmissible to another person or a risk more broadly than within that individual.
Okay.
Sorry, just another question, Caitlin. So is the reason why Tropicalis is got a higher mortality is because it has a higher mutation frequency that leads to drug resistance? Is that generally what's thought?
Good question, Julie. It's not something that I've delved into in this data. It's certainly, one of the, so there are a few reasons that a higher mortality can occur. And one of the biggest reasons is getting the wrong treatment.
Okay.
So if higher rate of resistance, a higher likelihood of unsuccessful therapy, but there are other papers looking into virulence markers as well.
Okay, yeah, I also wondered whether anyone had looked at mismatch repair mechanisms. So when I saw MMR in your talk, that's immediately what I thought that was, but then it turned out to be something different. But yeah, I mean, I just know from some work that Alex Idnam is doing down in Melbourne where he's looking at mismatch repair mechanisms. I mean, it might be that that kind of mechanism in Tropicalis is a little bit more leaky, perhaps. I mean, it would just be really interesting to look at from a basic science point of view, if that's really the cause of a higher mutation rate in this species.
Really interesting question, Julie, and it's just brought to mind a paper that did look at MMR and the fact that it's near-centromere on chromosome 5, which is the chromosome that has the ERG11 and TAC1 genes, which are important in azole resistance. Yeah, that's something for me to delve further into.
Thanks for an interesting talk.
Thanks for the questions, Julie. Do we have any more questions for Caitlin? I see no hands going up, so you may be off the hook, Caitlin. I'm kind of interested to know, like, based on the work that you've done up until this point and the things that you've found, what is it that you're most interested to look at next?
I'm glad to hear this question from you, Justin, because I know that one of your key interests is agricultural azole use. So I thought that that would link in with one of your research topics in Southeast Asia.
Excellent. Well, I see today they found Candida auris in wetlands in India. So now they're proposing that this is a source. So who knows where environmental interactions for Tropicalis might be found. It's really interesting.
Wash your fruit.
So
okay.
I have another question for Caitlin.
Okay, great.
How useful do you think then would the DST/MLST scheme be? Is it a tool that if, for instance, lots of hospitals did it because there's obviously the Sanger-based approach, which is a lot simpler. Do you actually think that if hospitals were to adopt this relatively cheap, if you like, screening tool, that it would actually assist in any way for triaging of patients or, changing outcomes, I guess?
Yeah, interesting question. I suppose I'd like to invite a comment from Sharon and Sebastian and others, but I would think that what's probably more useful clinically is a rapid marker of resistance. So given that a lot of those clusters are associated with OG11 mutations, directly looking for the OG11 mutation as an early marker of likely very high Azole MIC would be useful. And there is a lot of work going into that using sloppy molecular beacons, for example, to detect mutations in the OG11 gene. I know, Kate, I can see you online and you've done a lot of reading around that as well. MLST, other than that, in the clinical setting, I suppose would enable a rapid identification of an outbreak if there was one. And if there wasn't access to whole genome sequencing while we're waiting for it to get cheaper and more streamlined. Is that along the lines of what you were thinking? I know that you've done a lot of thinking about this as well.
Yeah, no, that's what I was getting towards. Thank you, Caitlin.
Thank you.
Thank you, Caitlin. I saw Caitlin was inviting comments from either Sharon or Sebastian there. I'm not sure if either of you had something you'd like to add.
I didn't mean to dob you in.
Perhaps not.
No,
Oh.
nothing to add. Sorry, struggling to find the unmute button.
Yeah, causes delays. Hey. All right. Well, in that case, there's also no questions in the chat, so just remains to say thank you very much, Caitlin. That was a very interesting talk. Good to hear what you've been getting up to. We'll be having another talk from Fungisphere next week, next Friday, from Tien Huynh. So hopefully we'll see some of the same faces for that. Look forward to it. So thanks again, Caitlin, and thanks everyone for coming. We'll look forward to the next time.
Thanks, Justin.
Thank you very much for the opportunity and for the excellent questions.
Okay, great. Thanks.